I am Julie Clark. I am the Vice President of Clinical Development at Iveric Bio, and I'm very excited to kick off our Spotlight on Dry AMD session. So avacincaptad pegol is a pegylated RNA aptamer that was specifically designed to inhibit complement factor C5. I am here today to talk about the two positive pivotal studies, GATHER1 and GATHER2, that make up our dry AMD program. Both of these studies were well-controlled, double-masked global studies that enrolled a representative patient population and evaluated a clinically meaningful endpoint. This was actually part of a special protocol assessment with the FDA. Clinical safety was also comprehensively evaluated in this program. GATHER1 evaluated 3 doses of avacincaptad pegol, 1, 2 milligrams, and 4 milligrams. These were dosed monthly via intravitreal injection and compared to sham, also dosed monthly. The primary endpoint was mean change in GA area from baseline to month 12. The study continued in a masked fashion out to month 18, allowing us to present those descriptive analyses. GATHER2 randomized patients in a 1-to-1 fashion to the 2 milligram dose of avacincaptad pegol versus sham, also delivered monthly. The primary endpoint again was at month 12. This time we evaluated the mean rate of growth, so the slope analysis in geographic atrophy, and this was actually part of the SPA agreement with the FDA. After that month 12 endpoint was evaluated, patients that were initially randomized to the active arm were then re-randomized to continue to receive monthly avacincaptad pegol or to extend that dosing interval to every other month. Those patients that started on sham completed the study on monthly sham therapy. The patients enrolled in the study had to have geographic atrophy that in part fell within 1,500 microns from the foveal center. However, that had to be non-center point involving. Patients with evidence of CNV in either eye were excluded from the study. This is an illustration that helps to show the types of patients that would have been included or excluded based on that center point involvement evaluation by the Duke Reading Center at the time of screening utilizing multimodal imaging. The injection fidelity rate was high in both studies, over 85% in GATHER1 and over 90% in GATHER2. Baseline characteristics were well balanced between both arms in both studies. So, the pre-specified primary endpoint was met in both studies at month 12, highly statistically significant reductions in growth. When we look at this treatment effect utilizing a different efficacy endpoint, utilizing the observed GA growth data, we see consistent results. Both studies, month 12, highly statistically significant reductions in growth. When we look at treatment effect across the predefined patient subgroups, again, we see the treatment effect consistently favors avacincaptad pegol treatment versus sham. GATHER1, month 12 results, GATHER2, month 12 results, and GATHER1, month 18 results. Now let's pivot into safety. It's worth taking a minute to remind ourselves that aptamers are small molecules and they're synthesized through a chemical pathway. This is not a biologic. There is no biologic intermediary, and this may be advantageous as it prohibits the opportunity to introduce some of these immunogenic contaminants into the process. Patient disposition was similar across both arms in both studies. Ocular adverse events were more frequently observed in the eyes that received active treatment versus those that received sham. And when we look at the serious ocular adverse events, these were low and similar in both the treatment arms, avacincaptad pegol active treatment and the sham arm in GATHER2. When we look at those common ocular treatment emergent adverse events, those that occur in greater than or equal to 2% of the patients, No surprise, a lot of these are associated with either the preparation or the injection procedure itself. We'll talk about CNV in a second, but the other event to sort of take a look at more closely are these IOP increases. These were largely post-injection, they were transient and mild and resolved the same day. Please remember, this is a 100 microliter active injection. When we look at serious ocular treatment emergent adverse events, again, low across the arms, there were equal numbers of serious ocular events in the avacincaptad pegol arm and the sham arm in GATHER2. There were 2 cases of choroidal neovascularization that were deemed serious by the investigators in the active arm, and 1 case of CNV that was deemed serious by the investigator in the sham arm. There was 1 case of mild vitritis in the first year of the GATHER1 study. Otherwise, we did not see cases of inflammation, endophthalmitis, or ischemic optic neuropathy. Now, CNV. In the GATHER1 study, patients that were found to have developed CNV were exited from the study. In GATHER2, if a PI suspected CNV, that patient was fully imaged, OCT, FA, FP. Those images were evaluated by the Duke Reading Center within 1 hour and confirmed the diagnosis. Those patients continued in the study. They continued to receive IP as well as on-label anti-VEGF treatment. We've started to look into these data with our great partners at the Cleveland Clinic, and they have determined that the majority of these cases were exudative in nature. So here we are. We have 2 positive phase 3 studies showing consistent treatment effects across primary endpoints and other efficacy endpoints, as well as patient subgroups. We also see a consistent safety profile across both studies. We're excited to be leaders in this space. We have submitted the clinical portion of the NDA exactly 1 month ago on November 3rd. We've also achieved breakthrough designation from the FDA, and we're working very diligently through that regulatory process to bring this therapy to patients. We're preparing the team, we are expanding, and we're able to really attract some of the top talent, some of the best people we've worked with across our careers to be able to bring this therapy to patients. So I just want to say thank you, particularly to all our patients, all of our sites, all of our investigators, and everyone at Iveric working very diligently right now. Thanks so much.